Chapter 9, Cell Cycle and Cell Division. Okay, so learning objectives for this chapter, by the end of it, you should be able to illustrate the cell cycle. And the cell cycle includes three separate phases to it, which within those three phases, there are more phases. So we have interphase, mitosis, and cytokinesis. Be able to describe the steps in mitosis as well as interphase, know the difference between a cell and a stem cell, and learn how regenerative medicine is being used to replace damaged tissues and organs. Okay, so let's start off with some of the things that kind of lead into cell division. So you know that we have different tissues in our body. We have nervous tissue in the brain, we have connective tissue all over our body, we have skeletal muscle, epithelial tissue, that's like your skin, cardiac muscle, your heart, smooth muscle in, say, your intestines and some of your digestive tract. And basically all of these tissues are, or let's just go over what a tissue is. So a tissue is an organized group of different cell types that work together to carry out a particular function. So this group of cells is basically performing a similar function. That's what a tissue is. An organ, on the other hand, is composed of at least two different specialized tissues. So keep those in mind when we get into talking about some of these regenerative medicine things and we're talking about creating organs out of different tissues. A tissue is an organized group of cells that have a similar function. So you have connective tissue, cardiac muscle, but you have both of those things in an organ, right? So like in our heart, we have both connective tissue, skeletal muscle, cardiac muscle. Well, we don't have skeletal muscle, sorry. We have connective tissue and cardiac muscle, as well as some smooth muscle, actually. So we have a few different types of tissues that go into making an organ. So how, you know that as you grow up, as you grow older, and if you get hurt, that your body is, for the most part, able to repair itself. And one of the ways that it's doing that is cell division. So that's just the way that our body or our cells reproduce themselves. So why do normal healthy cells divide? Well, there's really ultimately three reasons. The first is growth and development, right? We start out as a little embryo. Even before that, we start out as an egg that's fertilized by sperm. Specifically talking about humans. Obviously, this goes for many other organisms as well. They go through similar growth and development. But that's one reason why normal healthy cells divide, is to grow and to develop further into whatever organism they're becoming. Again, we'll use a lot of human examples in this, especially since this chapter focuses a lot on regenerative medicine. The other is cell replacement, and last but not least, we have healing wounds. So just to go into these a little bit more, growth and development, like I said, you have a fertilized egg, and then it just continues to grow and grow and grow. When we're talking about mitosis specifically, which is the bulk of this chapter, later we'll get into something called meiosis, which is different and involves sex cells. So mitosis is your body cells, okay? So you'll hear it often referred to as somatic as well. Somatic just equals body for the most part. So these are your body cells. So we start with a fertilized egg and then we divide that, well, not divide it, sorry, we replicate it into daughter cells. When you hear the term daughter cells, it has nothing to do with sex. We wouldn't call them son cells. Daughter cells just means that it's two identical cells. So keep that in mind. It has nothing to do with sex. It's just daughter cells are identical cells, okay? We'll write it out here so you guys got it. Okay, so our fertilized egg and the daughter cells just continue to divide and divide and divide and replicate until you have trillions of cells that make up the human body. Eventually, we get into a fetus, then we grow into, eventually all the way into an adult human. The second reason that our normal healthy cells divide is cell replacement. And cells are being replaced pretty often, right? Not all the time. Most cells do have a finite lifespan. Some of them last longer than others. So cell division is happening pretty regularly within your tissues, and it's doing so to replace dying cells and maintain healthy tissues, basically. So cells in the bone marrow regularly divide to produce new healthy blood cells, and then, they kind of work their way out. There are some cells that basically never go through mitosis, like our nervous tissue, our nervous cells, neurons that we have in our brain, those are pretty much never replaced. I think your eye lens or the lens that's within your eye is never replaced. And then other ones go through pretty quick turnaround. So like our skin is being replaced every couple weeks. Red blood cells, maybe every few months. Bones take about 10 years. When you break a bone, right, it takes a little while to get it back. But these are things that are still happening, and it's one of the reasons why our cells are dividing is to replace other cells that we need to in order to maintain a healthy system or a healthy organ. The last reason is to heal wounds, and I'm sure you all have seen this before. Any sort of injury that you have really triggers cell division to replace those damaged cells, so you can see what's happening after 6 hours to 1 day to 2 days to 7 days. And we do have scars, and we develop scar tissue sometimes, right? But ultimately, our unless you have something wrong with you, there are things that can go wrong, or it can prolong your healing process. But for the most part, our cells divide pretty quickly, especially in something like the skin, to be able to replace the cells that you lose in that injury process and hopefully heal up pretty fast. And again, it might leave behind a scar, but for the most part, we're able to heal relatively well from more superficial things like this. Okay, so now that we know why cells divide, how do they divide? Cells don't simply just split in half, right? If they did, they would just keep getting smaller and smaller and smaller, and eventually you'd have basically nothing in these cells. So they don't simply split in half. They actually have to go through a series of phases that we refer to as the cell cycle. So it has to go through a preparatory phase, and then it goes into a division phase. Okay, so it's an ordered sequence, and keep in mind when this is happening, cells are throughout your body are in different phases of the cell cycle. And it does go within the cell cycle. And for the most part, the uh, preparatory phase takes a lot longer. Say it takes a whole day or 24 hours to go through all of cell division. You would spend about 22 hours of that within the preparatory phase and only 2 hours in the division phase. So regularly when you're looking at cells, say under a microscope, the chances that you'll see it in the preparatory phase, which is also referred to as interphase, which we'll talk about here in 1 second, are great. So you will probably see more cells in interphase than you will in any sort of division phase within mitosis or cytokinesis because they spend so much more time in that. Okay. So let's get a little bit deeper into the cell cycle. So we know the cell cycle, again, the cells aren't just splitting in half, they're going from one cell into two identical cells, which we often call daughter, right? That's what we went over a little bit at the beginning. Okay, so first off, we're going to start off with our preparatory phase. And the preparatory phase is just like it sounds. We're prepping to be able to divide. And since we're not just getting smaller every time, we've got to copy all of those cellular contents. So we've got to duplicate all of the organelles, the DNA, and the cytoplasm. Remember, each new cell has the same amount as the original cell, so you've got to double everything that you originally had. This phase collectively is called interphase. It does have 3 different phases within it. Okay, so we have G1, S, and G2. So a lot of times when we're specifically referring to interphase, we might abbreviate it I and then say which part of that phase. So if I ask you something specific, like in which stage is DNA replicating, you would tell me interphase, but you would also tell me the specific part of interphase, which would be S. We'll go over that in just a second. But just keep that in mind if you see any questions or if you do the Chapter 9 class activity. Interphase is usually not specific enough. Please tell me which part of interphase you're specifically looking at. Okay, so in interphase, we have G1, S, and G2. That is all of our preparatory phase in total. And G1, our cell is basically getting nice and big. So we have our regular cell here, right? And now it's basically going to double. It's making lots of extra cytoplasm, getting ready to prep everything and duplicate organelles and everything so that it can actually divide eventually. Once we get into the S phase, the main thing that happens here is our DNA replication occurs. This is where we're duplicating all of our chromosomes. And you can see, you can follow along the chromosomes here. So we actually have, so a chromosome, just to kind of get some of this lingo out of the way because it gets confusing. So let's say our chromosome is here. I usually draw it as like an X just to be easy, but it's really like one chromatid here, one chromatid here. Okay, so a chromosome is made up of two chromatids, right? So when we're looking at this, you can see all of them right here. So we see our two red, our two green, our two blue, and then in S, we're actually going to duplicate this, and we're now producing two identical sister chromatids. Okay, so sister chromatids mean that they're identical. Normally, we do not have sister chromatids except for in this phase. So sister chromatids mean identical. Okay, so that's happening during S phase of interphase. Again, we're producing identical sister chromatids. Then we go into G2. That's like our final preparation. So anything that hasn't happened, taking a little bit of extra time, just making sure that everything is perfect and ready to go into the cell division or into the division stage of cell division. Okay, so it's just preparing every last thing that it can. Okay, now we're getting into division. So we went through all of our prep and interphase. Now we're going into division, and division has two main steps. First being mitosis, second being cytokinesis. Within mitosis, there's a few different phases. Okay, and we'll talk about them in 1 second. But the main things to know about mitosis is our chromosomes are being evenly divided. Our sister chromatids are being separated, right? So remember we have, like, say these are both red. They're now each going to go to their own cell. So we'll have one red in one cell, one red in the other. Let's draw two cells. So we don't have identical sister chromatids in each cell. We're dividing them up here. And those sister chromatids are attached at what we call a centromere. So when you see them there, it's like they've got this little thing in the middle called a centromere. Oops. That's holding them together. Okay, and we'll go through the stages of mitosis here in just a second. So that's kind of the actual like dividing phase. And then cytokinesis is like when you take the chopping block and you cut them in half. So that's when our enlarged cell is actually splitting into two cells. Each cell has its own full complement of DNA. It has all of the organelles. It has all of the things that each of the other cells have. So you're going from one parent cell into two daughter cells. Okay. Okay. So as a general, when we're looking at this, we'll kind of go through like what the chromosomes look like in each part of the cell. So we'll start with interphase. Again, interphase, we have G1, S, G2. Okay, let's make this more of a 1. So again, G1, we're making more cytoplasm. S, we're making DNA. G2, we're doing all of our final steps. So when you're looking at interphase generally, if you see some cells that are in interphase, the chromosomes look pretty loosely gathered in the nucleus. So you kind of see them all jumbled up and gathered around. They're not well organized in this phase. Once we get into mitosis, mitosis has a few of its own phases. So here's where we're starting mitosis, where we're starting our division phase. And the first phase of mitosis is prophase. So this is where our chromosomes are beginning to coil up. So they're beginning to look more like what we consider chromosomes to look like, at least in eukaryotic cells, right? So our chromosomes are beginning to coil up. Next, we're going into metaphase. This is kind of an easy one to remember if you think of meta like middle. This is where our chromosomes are aligning in the middle of the cell. And if you went back, you'd see we have a nucleus, right? We have a little nuclear membrane surrounding it. And that's basically dissolving, and dissolving from that we have, or dissolving even just in general from our cytoskeleton, we have all these little microtubules that are coming out of it. And that's what's forming all these little spindle fibers that you see here. So these are attaching to the centromere right in the middle of our sister chromatids. Okay. In anaphase, what happens is those microtubules shorten up. So they go from here and then they kind of contract back and they go to each pole of the cell, basically, separating the sister chromatids. So the way I think about this, you have the centromere right here in the middle, and we have our sister chromatids here. And basically, these little microtubules are pulling on these. So it's pulling this guy this way and this sister chromatid this way. And eventually you're going to have, again, one in each cell. Again, this is really helpful to look at animations for this, which your book has. YouTube has some great examples. I'll try to post some. But overall, hopefully you're getting kind of a general idea of what it looks like. The animations are really helpful. And keep in mind, if we were in lab, you'd see this in some of the cells, but it's hard to see. You can see here in our animation, it looks really clear, right? And when you're looking at animal or plant cells and you're looking for different phases of mitosis or cell division in general, it's not super clear cut, right? Metaphase is one of the easiest ones to see because everything's just lined up in the middle and it looks nice and pretty. Anaphase is also fairly distinct, but it's hard to look at this and be like, oh yes, clearly that cell is an anaphase. So it does take a little bit of imagination when you're looking at these things. So really visualize what's happening with the chromosomes and mitosis really overall, this division phase of the cell division is really referred to as a dance because if you watch it in a sped up version where you're watching the whole cell division take place in a single cell that's forming another cell, the forming into two daughter cells, it looks like this really cool dance where these chromosomes are just dancing all over the cell, eventually becoming two daughter cells. So mitosis is often referred to as a little bit of a chromosome dance, and there's quite an art to it. So anaphase, our microtubules shorten, we're separating our sister chromatids, gathering the sister chromatids basically at each pole of this really big cell that we've got going on here. Last phase of mitosis is called telophase or telophase. This is where our daughter cell nuclei are formed. So you can see our nuclei forming here. They're still like pretty loose. It's not fully formed, but for the most part, we've got them going on here. And you can see that it looks like it's just about to divide, but it's not quite there. That's going to happen in the very last step of cell division, which is cytokinesis. That's where we're just like chopping it straight in half. Our cytoplasm's dividing, and then we're going to end up with two daughter cells. Okay. So that is a very kind of quick and dirty overview of cell division. That is happening with all somatic or body cells. So any cell that you are dividing and it's making an exact replica of the first cell, that's what we call, or that sort of cell division is mitosis. Again, meiosis we'll get into pretty soon. That has to do with sex cells and is a little bit different. Goes through some similar phases, but is still very, very different. So mitosis is happening with body cells. Again, we're taking one parent cell, turning it into two identical daughter cells. And, stuff does go wrong. Stuff does happen here, which we'll get into some of these like mutations and things that can happen. But for the most part, that's exactly what's happening. And again, our body is constantly replacing cells, right? And this isn't just happening in humans, it's happening in other animals, organisms, plants, all sorts of different things. But it's not uncontrolled, which we'll get into next chapter, I believe, when we start talking about cancer. That's what cancer is. It's uncontrolled cell division. Right, so we don't want this stuff just to be dividing constantly over and over and over. We want to make sure that we have the right amount of cell division because if our cells just kept dividing in all of our different tissues, our body wouldn't be able to support that. We would be huge, right? Because all of this stuff can take place in a relatively short time scale. So kind of going from that cell division, now we're going to go into kind of how your book frames all of this and get into a little bit of what regenerative medicine is. So regenerative medicine is using your body's unique ability, or it's not really a unique ability, it's an organismal unique ability to regenerate, to divide cells and to make more, to stay healthy, right? So scientists have been trying to grow human organs, which is both easy and not easy, depending on which organs you're looking at. Again, organs are just two or more specialized tissue types. But the human body is constantly regenerating, though not all the time. And we have different types of cells, right? So one that you might have heard a lot about and is really, really important in regenerative medicine is stem cells. And stem cells are just like, they're awesome, right? These are our immature cells that divide and then differentiate into specialized cell types. So these are like the ones that haven't decided what they want to be when they grow up yet. So these are the ones that can be anything they want. They have such a bright future ahead of them. So these are why they're super important, why we have a lot of stem cell research, why a lot of regenerative medicine focuses on stem cells. And you've probably heard stem cells and science in general. A lot of times stem cells get a really negative or bad rep because a lot of people think that you can only get them from embryos. So a lot of people think that we abort babies for stem cells, blah, blah, blah. There's all sorts of reasons. And the truth is, adults even have stem cells. Everyone has stem cells. Baby stem cells or embryonic stem cells are really valuable, but it's not something that you're usually purposely aborting babies for. So there's a lot of misconception with that. Even if you see people practicing regenerative medicine, I know about some regenerative medicines that are used from volunteering mothers. And it's not from an aborted child or anything. The child's stillborn, is super healthy, everything. But you can get stem cells even from like amniotic fluid from the mom, from the placenta after birth. So you can get stem cells from all sorts of different places, including adults. Okay, so don't think that stem cells always have to have a negative background to them. Stem cells, again, are just this immature cell that can differentiate into more specialized cell types. So they are really, really valuable for multiple reasons. Okay, so a few different ways we can get into regenerative medicine. Specifically, your book talks quite a bit about bladders, because bladders are honestly pretty easy. They're only really made up of two sort of specialized tissue types, which is epithelial tissue, which is like the inner lining of the bladder, and then the muscle tissue, which is the outer layer. And again, epithelial is kind of, you can think of it almost like skin. It's kind of like the um, skin is your epidermis, but it has an epithelial layer to it as well. So you take a diseased organ like a bladder, right? And it only has two different kinds of tissue types. So you can remove it, and take a healthy portion of the patient's bladder with the two main tissues with it, divide the cells in culture so we can put them on a little petri dish, have them go through their own cell division, um, and then add those cells to a biodegradable scaffold. And this is where they form tissues to basically make a functional bladder. And then from that, you can take that engineered tissue and transplant it back into the patient. Patient outcomes are really good, improved urinary function, no immune rejection because it came from the patient's own cells. So keep in mind, anytime you have the option for anything like this, whether it's regenerative medicine or any kind of surgery or anything like that, it's always beneficial to generate an organ or to use your own stem cells because the chances of rejection are greatly decreased because the cells are from your own body, right? So your cells recognize your cells, and even if you have similar plasma, blood type, blah 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 blah, as someone else, like say you're trying to give your kidney, one of your kidneys to someone, that does not ensure that they will not reject it. Okay, so there's still quite a benefit to having from using your own cells. And again, our stem cells are the ones that don't know what they want to be when they grow up yet. And they have to go through this process called cell differentiation. And cell differentiation is just the process in which a stem cell develops into a more specialized cell type. And to do this, it has to be given what are called growth factors. So something has to signal that cell and say like, hey, we need some more muscle cells. We need some more smooth muscle. We need a specialized muscle cell. We need a specialized B cell. Whatever it might be, if those growth factors are around, they're basically saying like, hey, we need some help in this area. So our stem cells that don't know what they want to be all of a sudden have this awesome career being, let's say, a specialized muscle cell. So they go into that. Okay, so that's why stem cells are really, really valuable and how they go through cell differentiation in a very broad way. So when we use these stem cells, we can use different sorts of medicine, right? One is therapeutic drugs, right? And we can use these to stimulate some cells within the body to repair damaged tissue. So we can put specific growth factors in to differentiate stem cells. So we can have growth factors that are processed into therapeutic medicines that you take orally, and then the patients can take that and it'll stimulate some cells in the brain to differentiate into nerve cells and repair damaged tissue. Because again, our nerve cells really don't regenerate. They're never replaced, which is why we go into lots of things like dementia, Alzheimer's. The longer you live, eventually your nervous function, your brain function really becomes greatly decreased. Even if it takes a long time, eventually you'll get there. So therapeutic drugs where you're adding growth factors to tell your stem cells to develop into, you're obviously adding specific growth factors for this to happen. And then you can kind of differentiate those into nerve cells to repair that damaged tissue that you are experiencing. And therapeutic drugs have a lot of major benefits, one of them being obviously that it's way less invasive than any sort of surgery or injection. So if you can take oral therapeutic drugs, those are great. We also have biodegradable scaffolds, and we can use these to grow tissues and organs outside the body and then transplant them to replace damaged tissue, which is pretty cool. So you kind of do the same thing. You have stem cells that you isolate, you put into a culture to go through some cell division, and then you add some growth factors, layer it on a biodegradable scaffold. They begin to differentiate into a functioning tissue, and then you can transplant that tissue into the patient. Ears are another one that's pretty easy. Bladders are pretty easy, mostly because they're only made up of two tissues. If you get into something like kidneys, kidneys have a lot of different specialized tissue types involved. Also, the blood flow within something like a kidney is really, really intense. So there's lots of different blood flow, lots of little capillaries going on, and it becomes really hard to manufacture that kind of organ outside of the body. Ears, I always think of this crazy picture. They did it on the back of a rat, and you can look it up. It looks pretty intense. You've got like this rat, and he's just got an ear growing on the back of him. He's serving as the biodegradable scaffold. So that's interesting. The other thing that you can do is you can do injections. So you can stimulate these stem cells outside of the body, add some growth factors, and then you can inject them into places like, say, the heart if you want to differentiate into some healthy cardiac muscle to be able to repair any sort of damage you have in your cardiac cavity or in your heart. One of the last options is cadaver organs. And probably sounds kind of strange, right? A little bit Frankenstein-y, maybe. But basically, you can take a cadaver organ, like say a kidney, and you decellularize it. So you use a mild detergent, you wash it up, any of the cells, basically anything that is attached to the old human it came from, or organism in general. And then you can inject some stem cells from the patient that you're going into, right? And now that regenerated organ can be transplanted into the patient. And as long as you decellularized it really well, you do actually have your own stem cells in that organ. So it's a really nice way of being able to manufacture an organ outside of the body by taking it from another body, from a donor, and put that into you while still probably having less of an immune rejection just because you do have your own stem cells going into it. So that's a little bit about regenerative medicine. Honestly, the US is not as far in regenerative medicine as some of the rest of the world, because some of the rest of the world is a little bit less regulatory in those things. And, you know, there's good and bad associated with that. I think Europe is probably higher up in regenerative medicine than the US overall. And again, there's good and bad things associated with it, and it depends on what you're doing and what you need done. But there's lots of cool research going on within regenerative medicine if you're interested in that. But overall, if you're doing the Chapter 9 class activity, it's going over the cell cycle and modeling the process. So you can, for part 1, you can just indicate again what sort of phase it's in. So again, if it's interphase, you can write out I and then tell me whether it's in G1, S, or G2. There is, I will say, give you a hint that there is one of these, I'll tell you that it's the last one where chromosomes are loosely gathered, not visible as distinct units. That is actually just interphase in general. Any phase of interphase that you're looking at, that's kind of what the chromosomes look like. And eventually, right, you have more and more chromosomes once you get into S and you're duplicating the DNA. But generally, that's kind of what's happening. And then with mitosis, you can obviously list out any of the phases. So mitosis, we start with what? So mitosis, we're starting with prophase, going into metaphase, then anaphase, and then telophase. I'm sure there are good mnemonics to remember this. I just remember PMAT. So prophase, metaphase, anaphase, telophase. And again, prophase, prophase is where you're kind of breaking up the nuclear membrane, but we still have our little sister chromatids here, right? And then metaphase, we're going to line them all up. And we're taking this nuclear membrane here that we have and forming those microtubules, right? And then anaphase, we're going to kind of pull them apart towards the other end. And then telophase, we're kind of looking just like we were in prophase. So kind of looking the same here, but now we just have two of them, right? And pretend this has four, let's say. So now we just have two of them. So prophase and telophase actually look kind of similar. And then cytokinesis is the very last one. I'll just say that's a C. And that's where we actually have two different daughter cells, okay? Each with its own little nuclei in it. I know this probably looks like some weird face that I'm drawing. So please keep in mind that generally when you're talking about interphase, you should specify which phase of interphase. The same with mitosis. Mitosis has very different phases within it. In general, we have our preparatory phase is interphase, and then our division phase is collectively mitosis and cytokinesis.